Hello students, welcome to Legacy IAS Academy. In today's video, we are going to discuss about a very significant project that India has successfully completed, that is the Genome India Project. So we are going to discuss what this project is all about, what do we understand by the term genome, and also how this has helped in creating a genetic map of India. So first of all, to give you the context of this particular issue, obviously the government's Genome India initiative, which has announced its successful sequencing or completion after sequencing 10,000 whole genomes of healthy person that has been collected from all across India. It thus has helped in creating a genetic map of entire India. In this context, researchers from various eminent institutions, especially science institutions across the country, they were the one who has helped in collecting the blood samples, then sequencing the genome from those blood samples, and then developing a methodology and further storing such a huge volume of data that has been generated here. Since we are talking about data, let us try to understand that how much data has been collected and where this data will be stored or how this data will be used. So obviously each sequence requires almost 80 GB of storage space, and since thousands of genomes have been sequenced, the collection of huge data sets of almost 8 petabytes is something that has been gathered. Now, obviously, such a huge amount of data, which is also very sensitive and important data, it will be stored at the data center, which is called as IBDC, Indian Biological Data Center, located in Faridabad near New Delhi. Now, how these data will be used? So, in this context, the government has said the data set will be made available to the researchers as a digital public good. Now, digital public good means basically researchers, if they want to utilize this data largely for medical study or medical research, they will not have to pay any kind of fees or users fees. Rather, it will be used, it will be given to them in a free. And that is why what we mean by the term of digital public good. Then data that can be utilized to develop new diagnostics, targeted therapies, identification of new rare diseases, and also the existing one is something that will be helped by this particular data collection. Now let us try to briefly understand about the Genome India project. So it was approved by the government in 2020 with the aim of creating a comprehensive catalog of genetic variations that is found across the Indian states. The rationale behind sanctioning of subjects lies in the fact that this kind of project, this kind of collection of such a huge volume of genetic data will be essential in understanding the history of the evolution. We know that in India we always have dispute that which ethnic groups came first, which ethnic groups have intermingled with which ethnic groups. So these are something that can also be answered by studying of the human genome. Second, we know that many diseases which specifically are genetic diseases that will also be helpful in discovering of such kind of genetic diseases, that why these diseases occurs, what kind of mutations that happens in the genes of humans that can create certain kind of issues. And third, obviously once we are able to identify the genetic basis of diseases, obviously we'll be able to help also the it will also help in creating the therapies, creating the medicines, creating the diagnostic studies for the such diseases in the future as well. This cannot be done using the data that is available in the existing international databases. Obviously we have the Human Genome Project that was done at the international level, but the problem is that in Indian context its relevance will not be that much significant, because Indian genomes are likely to be different from that of the other populations where a study already has been done. So researchers who analyze almost 5,750 of the genome sequences have already identified 135 million genetic variants from the Indian population. Now, if talk about the genetic variation, since the term is genetic variation, we're talking about in Indian population, analysis of 5750 of genetic sequences have already identified 135, that is 135 million genetic variants that is found in India. Why it is so? Researchers argue that this is because our population, which obviously is very humongous, 1.4 billion population almost, consists of more than 4,600 distinct groups. And in this context, also, if you look at the history or the culture of India, most of these groups have been practicing a marriage practice or marriage type that is called as endogamy. Endogamy basically means marrying within a single group. So due to this, as the group or the individual of group is not marrying outside of its group, they are able to maintain their distinct genetic makeup by this process. Third, this can also help compare and contrast the impact of genetic variation on the physical health of the general public. And in this context, the Union Minister of Science, Dr. Jitendra Singh, has said that after looking at all these data, we can say that India is the largest genetic lab in the world with such a humongous nature of variation. Now, how genome and its sequencing is done? So first of all, we can try to understand what do we mean by the term genome itself. So in simple words, or in layman's term, genome can be described as a biological instruction manual that we inherit from our parents during birth. It is a tone written with just 4 letters, that is A, C, G and D, where A stands for adenine, C stands for cytosine, G stands for guanine and T stands for thymine. So adenine, guanine, cytosine and thymine, these are the 4 bases. When they come together, they create everyone's unique genetic makeup. And as we can see, the most well-known model of the DNA that we have, that is a double helical structure. First of all, it was discovered by Watson and Crick, for which they also got the Nobel Prize. So basically, this is the double helical DNA structure, and between these bands, as you can see, is we have the 4 bases. We have the AT pair, that is adenine thymine pair, and then we have CZ pair, that is cytosine and guanine pair. So these pairs are what makes up our genome. Now, complete human genome, if we talk about, it is around 3 billion pairs of these bases. That means this is one pair, this is second pair. So such kind of 3 billion pairs are found in a single human genome. This contains all the information that is needed to create the physical health of a person, or maintain the health of the person, or maintain the physical features of person, physical forms of the person throughout his life. For example, the height of a person, how tall a person will grow, that is determined by the genes or genome. Color of the eyes, genetic diseases a person can get, or those which a person or individual is at a higher risk for. All these information is basically encoded in this particular genome. So in one way, what we can understand, what is genome sequencing? So in simple terms, we can define it as a method of determining the entire DNA sequence of an organism's genome. In simpler terms, it determines the order of A, T, C and G, the 4 bases that we have discussed that make up an organism's DNA. A genomic sequence is depicted by a very long line of these letters arranged in a specific order, and thus we can create also that how the genomic structure will look like. If you go slightly in more detail that how the sequencing is done, so generally this is something that is used worldwide. In the first stage, the scientists extract the information from the blood. Then, with a complete sequence of 3 billion pairs being extremely hard to handle, scientists cut these pairs into small pieces and tag them. It's same like how you do disassemble the furniture. And then the ACGT, the 4 bases, the codes of these smaller chunks are written down by a DNA sequencer, and then the complete sequence is put together. Now, earlier time, since technology was not that much developed, it used to take a lot of time for completing a genome sequencing. However, as per the current technology, current science and technology, we can say the scientists argue that in a in a time period of almost 3 to 4 months, the entire genome sequencing can be completed. As we have seen in the case of India, the 10,000 different species, 10,000 different human genome sequencing that we have done, that was completed in a time period of only 3 to 4 months. Now, obviously, the last question that can come to our mind is what is the purpose of genome sequencing, or how does the studying the genetic makeup of a person can help, or not only persons, since we are talking about genome sequencing at the level of India, how does studying the genetic makeup of the entire country can help us, in what context? So let us try to understand this from the example. Now first, such sequencing and such data can help in identifying the genetic basis or genetic risk factor for various diseases. For example, if we talk about India's case, we have one mutation that is called as MYBPC3 mutation. This mutation in the genes is responsible for cardiac arrest that happens at a relatively younger age. Now, this mutation is not so much prevalent at the worldwide level, but it is present in almost 4.5% of Indian population. That tells us that India's population is much more prone to cardiac arrest at a young age, and this is something that we are also seeing in the present time, especially if you're reading news and all these kind of things. So obviously, most of the time we blame it on the lifestyle and other factors. However, genetic factors also plays a significant role here. The other example we can give is of LAMP3 mutation. In LAMP3 mutation, a lethal skill condition basically arises, and this is again seen in nearly 4% of the population that is living near Madurai, but it is not seen in the global databases. So this is an example of how we can identify the genetic basis or risk factor for the various population group. The other help that this kind of study can give us is in targeting treatments, especially targeted treatments that we do for the rare diseases that usually arise from the genetic anomalies. Third, such kind of database will help us in developing new therapies that can cure these diseases. Example, mRNA vaccine that has been prepared by the scientists of India to prevent relapse of pancreatic cancer. This was possible after studying of the genome or genome sequencing. And last, we can understand help in identifying resistance indicating variants. For example, genes that might make certain medicines or anesthetics ineffective in certain population. Then in that case, when the treatment is being done, doctors will be aware about which medicine one had to administer. For example, if you look at the population of Vaishya community from the South India, they actually lack the gene for properly processing common anesthetics. And that is why for this group, use of such anesthetics can result even in death. And this is something which can be considered actually as very vital information, especially when a person is hospitalized and surgery or operation is required. So this is how we can outline the importance that Genome India project will play in the overall healthcare or health infrastructure development of India. So that is all about this particular issue. I hope you understood about the Genome India project, its salient features, and the rationale behind it. Thank you very much.